Okay, so I have the video panel hidden right now. So just what I want you to do is, if you have a question, I'm opening up the chat box and you can type it in there, or you can unmute yourself, because what I'd really like you to do is ask questions. So I'm recording now, and what I'm going to do is open up the week 1 content objectives, and I'm going to start out by reminding you that the content exam is about what was covered here in chapter 1 and chapter 2, and then the weekly content objectives under chapter 2 in week 2 that cover the properties of water and biological molecules. What you're starting on this week with chapter 3, that will be on the next test. Okay.
And
question?
Then the content exam, when is that exactly? Because I'm looking and it doesn't say, and it's due on Wednesday, or
it's not due on Wednesday. It's due on Saturday night. And
is that the lab?
No, that
3 post quiz.
It should be in here under the to-do list, unless I forgot to add it.
I'm looking at the to-do list right now, so I'm a little bit confused.
Yeah, oh wow, it should be in there, because if we look at the online schedule, I wonder if I'm going to have to do a new share. Did the schedule pop up? I moved it over.
The schedule does have an exam for May 9th. Like, I've known that that's on the radar, but
yeah. So what I need to do,
it's not even in my list of things to do for the week.
Yeah,
so let's do that right now. Well, this is just well, we're recording. I'll write a note and I'll put it in. Okay. So you see this in purple right here. So it's due on the 9th. Maybe, I think what I didn't do was throw it in on Friday night when I was trying to get week 3 up and running, because there are a couple other professors who've been creating and editing this particular one, and so I didn't have access to it yet. But they finished it this weekend, and so I need to take a look at it, and I'll put an announcement up tonight and say, okay, add it to the to-do list. Remember, on the schedule, this is happening at the end of the week, and then that way everybody will be reminded.
Again, that was freaking me out.
No, I emailed somebody back like, look at the schedule, it should be there, and it's here in purple, because all those are in purple, right? And you saw, like, I bumped the coronavirus reflection 2 to be due this week and number 3 to be due later, just because everybody had so much to do with trying to figure out chapter 2 that I just shifted that one a little bit later. So the
coronavirus isn't due Saturday.
The number 2.
I thought that was due Wednesday.
Was due Wednesday.
So I have 2 up, and did I give you a number 3?
I didn't see one, so I thought only number 2 was due on Wednesday.
So we'll do number 2 is due on Wednesday, and I'll just edit this guy out in all of them, because you have enough to do.
Be like sometime next week or Saturday?
And I'll just make it due next week, and we'll just push it back.
Okay, cool. Thank you.
Yep. Okay, so I'm going to close the schedule, and then if I make my screen, this part, I'm splitting right now. I'm trying to get it to hide that so I can, every time I go to try to grab, uh-oh, I just accidentally closed it. I keep trying to grab it to make it big, and it keeps disappearing. It keeps pulling down the tab. So did you see my message where I had so much fun grading?
Yeah, that was insane. Thank you for doing it, though.
Yeah.
I'm glad you're doing so much. Okay.
Checking, like, to see if you posted a discussion, that takes me like 10 or 15 seconds because I'm just making sure you wrote something and responded. The whole purpose of the discussions is for you. Because you're talking to each other and you're helping each other. And so that's that's why I did them. My daughter is being funny. I'm really having a hard time grabbing this here. I'm going to do it like that. Oh, that was funny. It was behind the sharing thing, the little Zoom thing. All right. So if I increase the size of the text a little bit here, then I can highlight for you. So the first part of chapter 1 was on the process of science. Your chickens, Nora? You're going to be forever on video now, roaring like a dinosaur. So the first part of chapter 1 was about the scientific method, and I know we spent some time talking about inductive and deductive reasoning and hypothesis versus theory and independent and dependent variable and control variables. And I went through everybody's lecture assignment number 2, and with the exception of a couple people needing to be reminded, you know, how the graph should have looked, everybody did really great. I was really impressed. So did anybody have any questions about that part of chapter 1?
The graphing, meaning like the difference between like the scatter plot and the histograms and everything, right?
Yeah.
Okay.
So I'm not, you know, basically, I want you to understand how to graph
Okay.
and you know, recognize where things belong. But on a computer test, I can't really I'm not really going to have you graph anything, right? So if there's
I would hope not.
if there's graphing, it's just, or parts of the scientific method, do you recognize whether something is a hypothesis or the independent variable or the dependent variable? Look at the data. Can you figure out the conclusions? So it's using the process of the scientific method to analyze the information that's presented to you. So, you know, like a lot of people chose to do the scenario on the ants. If you got a scenario, could you interpret it and come up with and identify the different components? Does that help?
Yes, thank you.
Okay. And then with inductive and deductive, you just need to remember about, you know, deductive and inductive, what we're getting at as far as which one leads to making a hypothesis and which one helps you to make a prediction. And so that's, I'm scrolling down. I reordered things, and so my brain is remembering where it used to be in the old PowerPoint. Okay, so like slide number 12 and slide number 11 on that first PowerPoint are about inductive and deductive reasoning. And actually it's 10, 11, and 12. And then you've got theory is in 17. And basically, if I were to pop this open, it's just to remember that inductive goes from multiple specific observations to a generalization. So it's observation to theory. And then deduction goes from a broad theory to a specific hypothesis. So inductive is theory generating and deductive is hypothesis generating.
Cool.
And that's it in a nutshell.
Is this from chapter 1?
Yeah, it is. So there's just one PowerPoint from chapter 1. And so
Okay.
ideally, and I haven't gotten to the study guides yet. I was just trying to plow through everything else because I really want to take some time in the next couple of days after the lab is up to look over your study guides. The directions tell you to like take the weekly content objectives to inform the information that could be in your study guide. If all of these are answered in your study guide and then you create questions in the left-hand column, I know some people are emailing me and telling me how many hours that they're putting in, and I'm kind of surprised at how many hours it was. And I don't know, maybe you'll find you're over-prepared and you don't need to do quite as much for the next time around. That would be nice, right?
I wish.
Yeah.
So
So-
I'm just double checking because like, I mean, I do go back to the course objectives to ensure like that I'm understanding, but I do flashcards. I don't necessarily do it on the study guide. So
Okay.
like I now know, like, I mean, I know you're already looking for the Cornell style notes. That's how you wanted us to write our notes.
Mm-hmm.
I was just wondering though, like, I mean, you're probably just gonna like answer my question when you respond back, but I think I may have done them wrong because I did the Cornell styles, but I didn't use the course objectives to answer them. I kind of like went back on stuff that I didn't put on my note cards, like big stuff that I wasn't sure about.
Well, you need to think that I can't see your note cards. So
Mhm.
the directions for the Cornell notes tell you here, if I just pull open like the first study guide, I talk about, you know, the learning outcomes or the weekly content objectives and the PowerPoint to help deepen your understanding. And then right here it says use the learning outcomes as possible review questions in the left column and or guide your coverage of the content on the right hand detail side. So you wouldn't necessarily have to have all those, you know, every single one of those listed as a question on the left hand side, but if it's not covered in the right hand side, then you might be missing something that is going to be on the test. Now, I'll be honest, I'm not going to go through every single person's study guide and check every single item on that list. I'm looking for overall how's the format and what are you attempting to do? And for the first couple of them, I'm going to be giving pointers, and it's only if it's really, really bad are you going to lose points. And by really, really bad, I mean like you gave me a page for the whole chapter and that was it. There's no way that that's going to cover the depth and the breadth of the content that are on these content objectives.
Okay, so if you could just like leave me like a comment as to like how I can fix it, that'd be great.
Yeah. So I'll be doing that for everybody. I don't know if you saw the comments that you got. You know, you got a comment on the learning assignments.
Yeah, I saw those. Yeah.
I did have a question. If it's more than one page, how do we download it? I'm having a problem with that.
Well, if you typed it in Word,
Mhm.
it would be easy to upload it because then it's a multi-page document. But like it says in the resources in the orientation module, what you should be doing is if you're handwriting it, then you should be using a smartphone and creating a PDF using an app like CamScanner. There's no reason that you can't upload more than one page on there.
Okay, I'm not too good at it.
Well, CamScanner is just an app, and I've put the link in before, and you just have to download the app on your phone, and then basically instead of going to the camera on your phone, you would open that app and you can tell it to choose single mode or batch mode, and batch mode just means it knows that after you click the first picture, you're going to take more. And so then you just tap the button when you're done, and it lets you then click on the pictures. The other thing you can do, and I believe there's a link in one of these, or else I emailed this to someone and maybe I need to create a reminder post, was if you took a bunch of pictures on your camera and they're JPEGs, you can also go to a free, if you Google this, free online JPG to PDF converter. Then like 10 different website choices will pop up, and you can choose one, and it lets you drop your files in, and then it makes a PDF for you and it emails it to you.
Okay,
and so if you can drag files in the right order, then you can make a PDF using one of those websites for free. You want to watch a movie? Sister and brother
The
upstairs.
CamScanner scared me at first, but now it's just part of finishing an assignment. Then I take the pictures and then I submit it, but I was freaking out the first couple times.
It's something that my high school students do. I teach, and it's totally different. I do online high school, and I have some middle school students for just basic biology, where they are doing online courses. And for the company I work for, somebody else creates all the content, and sort of I'm a professional grader and advice giver. and that's the app that most of them use. And of course we know that, you know, just grab a fifth grader or an 8th grader or a 10th grader, and they'll show you things to do with your phone that you never thought possible, right? So the second part of chapter 1 was on the study of biology, and basically the critical pieces of that are the 7 characteristics of living things and the levels of organization, and then looking at taxonomy and phylogeny. So if I were to look at the PowerPoint here, that part starts with it starts with slide 31, where I talk about evolution. But that's sort of like the transition between theory, and evolution is a theory, to, you know, evolution basically explains how life has changed over time on the earth as we understand it. And so if you're going to go through this, slide 34 here has those 7 characteristics. So you want to be able to recognize, if you're given a scenario, like if I poked you and you moved away, that's a response to stimulus. Can you apply these terms to, you know, word problems that you would see on the test and answer them appropriately? And, you know, if there was a fill in the blank kind of a thing, it would be the same way, where you would have to fill in the right term that went with it. And this
How many
picture,
questions will be on the test? Because I know they're timed, but I just want to know like about how many questions there will be.
I need to look at it and decide how I like the questions. Basically, you'll have 1.5 to 2 minutes per question, depending on how hard the question is. The easier questions, you'll get 1.5 minutes. The harder
Okay.
questions, I would time it for like 2 minutes, and that's how I'm going to do the length of the test. I need to
Cool.
look at the questions and filter out any that I don't approve of, so that it looks, and I might add a few from my regular in-person test. My
Okay.
tests in face-to-face, I do as a mix of multiple choice and short answer and essay. And so I've just got to look and make sure that
And
this,
then like I'm not putting any pressure. I'm just like curious. Sorry if it seems like I'm pressuring you or anything. But like, do you know about like what time when the test will like open for us to take? Because I'm just trying to like schedule out.
based on the schedule, it's going to be open for 3 days. So you'll have the 7th, 8th, and 9th, Thursday, Friday, and Saturday to take in.
Okay. All right. Sounds good.
I'm considering making it through Sunday, but I really would like to think that you had a day of rest. And so I
good.
just want it to be done on Saturday night, so that you kind of go, oh, it's done, right?
Yeah.
And I had a
Um,
question. I had a question on the videos that you're recording for these Zoom meetings. I couldn't find the one from last time.
That is, so can you still see my screen when I make this big?
Yes. Mhm.
Okay. I first put it in an announcement, and then where I am popping everything in, I think because we're in my pre-shell, they're not over here. So let's go into one of the actual sections. I create most things in like a Canvas shell where nobody is, no students, and then I move it over. But under the orientation module is the Zoom sessions. And what I am doing, yeah. So
Okay.
what I'm doing is just dropping them all in there in order as we go. And one of the sections, even though I had checked it, the one from last week didn't immediately load, and I had to re-link it. I haven't heard anybody say it was still a problem, but yeah, here's starting from the first meeting, they go down.
And that's in the modules, right under the top of the modules?
So it's right after, so here's the orientation module. See,
Okay.
mine shrunk, and right between that and week 1, it seemed like that would be the easiest place for you to
Okay, that
find
sounds
it. And I didn't want to split them up everywhere in the weeks for you to go hunting for them. I just figured I'd drop them all into one place. Yep,
Okay.
that's where they are.
Thank you.
You're welcome. Okay, so back to content objectives. And then looking here. So know those characteristics and then be aware of cell theory, you know, that it's got these 3 components here, you know, and it sort of seems like a no-brainer. Well, of course, cells come from other cells, but when the first microscope was invented, we had to define what a cell was and what was a cell and what wasn't a cell. So, you know, everything scientific like that is based on cumulative observation. Okay, so moving forward here, most of these slides are about examples for the 7 characteristics. And then there's a slide 50 explains the domains, and the slide before that, 49, basically explains that, hey, way back a few hundred years ago, somebody named Carl Linnaeus was basically organizing species into groups. So he was before Darwin, and Darwin kind of built off of what he did, you know, trying to examine organisms and, you know, figure out how they were similar and how they were different. And so the most unique an organism can be is belong to the smallest group it can belong to is a species, where those are organisms that can successfully reproduce. And then whenever you see the scientific name of an organism, you notice that it has 2 words. The first word is the genus, and the second word is the species. So when we give our names as for, you know, human beings, it's Homo sapiens, and Homo is actually a category that more than one type of species fits into, and it just has to do with similarities. And so we're grouped based on similar structures and similarities in DNA, which makes sense. You know, how would you classify plants? You're going to classify them based on, you know, whether they're bushes or trees, and how do you know if something is a bush or a tree or a flower or a weed, right? It's going to be based on the characteristics and where you put the most similar things together. And that's basically how the entire system works for all living organisms. And I'm not having you, if you noticed, there was no detail in this, in the book or in the PowerPoint about having to name things. So you don't have to name things, you just need to know, you know, species is the most unique or distinctive, and the broadest is domains, and become familiar with what those domains are and what's grouped into them. And know that phylogeny just means we're organizing living and extinct organisms based on their similarities and their differences. And I introduced prokaryote and eukaryote in chapter 1, and we jump into that in way more detail in chapter 3, as some of you who've maybe gotten started on this have already noticed. And then the rest of this is just thinking about living organisms, and if we're going to study them, at what level are we studying them, and that basically informs, you know, this hierarchy informs us when we think about, if I'm looking at just a species, then I'm looking at organismal or population ecology. But if I want to understand how a group of individuals of a particular species interacts with other species around them, then I'm looking at community ecology. And if I'm interested in living in non-living parts of an ecosystem, then that's ecosystem ecology. And then when I'm looking at multiple ecosystems interacting together, that's landscape ecology. And if I'm looking at, you know, whole earth effects, then that makes sense that that's global ecology. That's all that slide was about. So feel good about chapter 1?
So.
Well, does it seem easy compared to atomic structure?
Yes.
We just start you off slow, and then all of a sudden the world's on fire, right? Okay, so let me pop these content objectives back over. And so my guess is, like, your first study guide would have been like a page or 2 to cover the process of science adequately, and then another page or 2 for the study of biology, that you didn't need to go on and on and on. And, you know, some people write more succinctly and they might have managed it in 2 pages, or if it was typed in 2 pages, right? And some
Sorry, I
people,
just had a quick question. Where do you find the content is like within the beginning of the chapter, right? Like that's what it is. It's like the chapter 3. It's let me pull it up. I know
these
it's like
content objectives are not in the chapter. The chapter is the basis off of which these were built, but that book does not have everything that you need in depth, and that is why you've got the PowerPoints and you have the videos to kind of take place of some of the lecture, you know, like direct speaking and sharing that I would have given to you, plus
Okay.
me doing the Zooms. So it's not that if it's not in the book, I don't need to know it. No, what do you need to know? This is right here. Use these to help you focus your learning and create your
Gotcha.
study guide. I'm
Okay.
not going back to the text, like I'll admit it, ever now. I know what's in there, and I don't go back to the text to write new questions. I base everything that I'm writing for a test off of these, the content objectives. And I use the PowerPoints and my instruction in person and the videos that I've shared with you that I believe cover these concepts in review and also in depth to complement the PowerPoint and the textbook. I want you to use all of those collectively. And that's why the direction for the study guide, especially the second one, where I said, this is how I really want you to do it. And explained, you know, how you'd read and take notes and then go back. Do you know what I'm referring to? The like study guide 2.1 and 2.2. So you got the basic instructions for Cornell notes for the first study guide. And then for the 2.1, the part 1 study guide, I gave you these directions and I re-explained using the learning outcomes and a PowerPoint, and use the learning outcomes as possible review questions and to guide your coverage. And then I gave directions for like, this is how you would do it. And so I put that in, I think, this one and then in the next one also. So it's right there as the directions. So if you're skipping the directions and just going to the study guide, think about how I provide you with information. I build this page in Canvas, and this is like me talking to you, except that I'm not there. You're pulling open my voice and reading it when you have the time to study. And so, you know, take this for what it says. This is exactly what I would like you to try to do. And it doesn't say how long the study guide needs to be. It explains how to go through the process so that you're layering or doing that deep processing rather than just trying to memorize facts or sequences. Okay, so we're going to go back to the week 1 content objectives. And we've got atomic structure, okay? And from the other Zoom videos that I have done where I talked about, you know, protons, neutrons, and electrons, talking about the periodic table, using the periodic table as a tool, that's all this part, okay? So, you know, basically you would need to be able to pull up a periodic table and have it available to you. And if you looked at carbon and you found that the number it had was 6, and here I'm going to open a tab and I'll just type in periodic table.
Can I ask a question? Can we use the periodic table on the exam?
Because it's an open resource at-home course, you can use anything that you have time to look at, but keep that in mind that part of the reason it's a timed test is
Oh, okay.
because I want you to have studied. Oh, this is going to
Yeah.
be one that isn't going to work that great. We have that back. Okay, I'm just going to leave it like this. So if I look at carbon over here on the right side of the screen, it's the C symbol and the 6 in the corner. You need to know, looking at it, that the 6 stands for the atomic number, and that's the number of protons, and that in the nucleus, and that would also be the number of electrons, because a typical atom of a given element is neutral, meaning it has equal numbers of protons and electrons. It only becomes unequal, like polar or charged, if there's uneven distribution of electrons in a molecule, or if it gives up or takes an electron and becomes an ion. And that this number down here, the 12.011, that's the atomic mass. And so what's 6 plus 6? It's 12. So most carbons have 6 protons and 6 neutrons, and so the average is pretty close to 12. But if you've ever heard of carbon-14, there are a few, you know, out of a pool of 1,000 carbon atoms, if I dipped in a scooper and I looked inside them, a few of them would actually have 8 neutrons instead of 6. And so my average atomic mass is slightly higher than 6 plus 6 because every now and again there's an isotope of carbon, meaning it's still carbon, it still has 6 protons, but because it has a slightly different number of neutrons, its atomic mass would be off. But what determines what element something is? The number of protons. And that's, so use the periodic table as a tool. You can look up any of this. And remember, I want you to focus on the 2 columns. So we can't actually see the left column over here. And don't worry about what's in the middle, but then worry about C3A, 4A, and then that's 5A, 6A, 7A, and you can't. I must not have clicked on a very good one. Here we go. This one, you can see it a little bit better. So you're focused on column 1 and 2 here, and then jumping over here, this is column 3A, 4A, 5A, 6A, 7A, and 8A, and really only the top 3 rows are biologically relevant. You have a little potassium and calcium in you, but the rest of this you don't find in a human body so much. So it's not really biologically relevant, so we don't worry about it for this class. So if you were going to build an atom of something, you have to put protons and neutrons in the nucleus, and you would need to have the same number of electrons in the electron shells. And therefore, if I was going to draw an atom of neon in E right here, and I see this 10, what would I put in the nucleus? I would put 10 protons, and then, wow, I really can't see that atomic number so good. I mean, that atomic mass. Here we go. This is a little bit better. My screen's kind of fuzzy. Okay, it's 20.18. And so I would just round down, and so I would have 10 protons and 10 neutrons in the nucleus. And then I would know that, oh, there's the same number of electrons because it's neutral. So I need to have 2 electrons in my inner shell because the first shell only fits 2. And then, well, what's 10 minus 2? It's 8. And so I would have 8 electrons in my second shell, and that's my valence shell because 8 fills it, and so I don't need a third one. And it might really help to think, hey, how many electron shells does an atom have? Well, if it's helium or hydrogen, what row is this?
1.
So how many electron shells?
1.
1. Row 2? How many electron shells? 2. Yeah, and it builds the same way as hydrogen and helium started. So the innermost shell holds 2 electrons, and so the valence shell, if you're in row 2, is whatever the atomic number is. So beryllium here, it's 4 minus 2. So there would be 2 dots in the first little ring around beryllium, and then 2 more dots showing the electrons in the valence shell. And so it has 2. And then if you think about it, how many does it need to be stable? Well, it's in column 2, so it needs 1, 2, 3, 4, 5, 6 to fill its valence shell. And in row 3, how many, because it's row 3, how many electron shells?
3.
3, right? And it needs 8 to fill it. So it's 2, 8, 8. And then see Kr krypton? See, its atomic number is 36. We jumped from 18 to 36. Well, what did we squeeze in the middle? All these transitional metals here. And so does it make sense that it takes longer to get from 19 to 36? And that by the time we get over to non-reactive krypton here, because its valence shell is full, it's got 4 shells, and its outermost shell or valence shell has 18 in it to be to be stable. Yeah, so it changes a little bit, but if you're taking chemistry, you'll eventually get to have all kinds of fun talking about s and p orbitals and the way electrons behave and molecule shape. But we don't go into that very much in here. We just worry about the first 3 rows pretty much, and that is in the PowerPoint on chapter 2.1, which was the chemistry review. So I'm going to come back over here to weekly content objectives. And so that's basically this first bullet point with its, sub bullet points in there. And also a little bit right here. We did this first one, describe the pattern by which electrons typically fill electron shells. And then this second one here that says, given the number of electrons in an atom's outermost electron or valence shell, predict the ion, if any, that will be formed, and then apply the concept of electronegativity to atomic behavior. One of the Zoom reviews, we actually went over that. And we were looking at one of the lecture assignments, and we were talking about that, and I had that electronegativity periodic table. Do any of you need me to talk about that a little bit more, or do you feel pretty good and you could go watch the video if you needed to refresh?
I
think
don't
we
know
should
number
keep
2.
Plugging through.
Just remind you where that was. Okay, so I'm going to pop open modules here and Zoom sessions, and I'll remind you which one that was, because I believe it has the image. See this one for April 27th? That one is where I had side by side the learning assignment and an electronegativity periodic table. And so I walk you through that. And then this one about Boseman covalent bonding, he really talks about electronegativity too. So if you're struggling with those, I would recommend you go back and look at that. And then this one, lab module 2, I'm looking at, actually I want to click on that one. Not that I want to hear myself talk. Actually, the electronegativity one is in this one, April 29th, and this one shows the regular periodic table. So you want the Boseman covalent bonding, and you want April 29th for electronegativity. And I'm going to click open the periodic table. There's a link in there. And if you were to click on that, I want to show you that not only is there that electronegativity periodic table that I used, but also there's a nice explanation right here about electronegativity that is complementary to what was in the lecture assignment. So, and then talks about the periodic table. Ooh. I'm going to click on that because look, there's a PDF, and that's really nice. So I'm going to remember to save that and put it in as a file. So which elements look hot?
On the right?
On the right, the red and the orange ones, right? So what do you think that's meant to convey in this periodic table? Look at the scale down here, green to red.
Like how acidic they are?
No, how electronegative they are. It's
Oh.
okay to be wrong. It's better to get your incorrect thinking out right now, right? Before the test. It's always better to ask or to say something and to be wrong than to never say it at all and then you don't know. So over here where it's, go ahead.
I had a question about the exam. Are you going to make us do any drawings on it?
No,
Good.
because it's multiple choice.
Okay.
So if you look at the green, see this, how it says low, and do you see 0.7, 0.79, 0.82, 0.98? And then as we move to the right, look at the kind of lime green, 1.36, 1.54, and then to yellow, 1.63, 1.66, increasing over here to the right, 2.01, 2.18, and then orange, 2.55, 2.96, and then red is greater than 3. So those numbers basically give a chemical electric measurement of how strongly atoms of a particular element pull on the electrons that are orbiting the nucleus. So remember, what's an atom? It's a positive center with neutrons that basically glue the protons together, because otherwise the protons would repel each other, because like charges repel. And that positive center has a pole or an attraction for the electrons, which is what keeps them in orbit around the nucleus. Sort of like there's a gravitational pull by the Earth on the moon, and also by the moon on the Earth, but the Earth is much bigger, so the moon is like in perpetual, gravitational pull falling around the Earth, but it never falls to the Earth, it just orbits, right? So the electrons, they don't fall into the nucleus, they orbit it, and they orbit it at specific energy levels. You can think of those energy levels sort of like the way planets orbit the sun. They're not all in the same lane, they're in different places around the sun. Well, in atoms, it works that way too, which is why we have the periodic table organized based on electron behavior and where those electrons are located most of the time. And so the smallest atoms are in row 1, right? Because they just have one shell. And then what happens to the atoms of these elements as we go down the periodic table? They get bigger and heavier. Their nuclei grow, but then what happens to their shells of electrons?
Multiply.
They multiply, and the valence electrons get closer or farther from the nucleus as we get more shells?
Farther.
Farther, exactly. So if I were to look at row 1 and I go from hydrogen, which is 2.2, and it has that one electron that's very close to its nucleus, and as I go down that row to CS, which is cesium, and FR, I want to say francium, do you see how the electronegativity declines as you go down from the top to the bottom? That's because the electrons in the valence shells of these bigger atoms are farther away, and so it's harder for those nuclei to pull on them and to keep them in orbit. And so electronegativity is basically a measure of that, an atom's ability to keep its electrons in orbit. And what happens as I go to the right on a particular row? We don't get another valence shell, but we get what? More protons in the nucleus. So we go from 3 to 4, 5, 6, 7, 8, 9, 10. And so why are we going from green to light green to yellow to orange to red here? It's because we're increasing the number of protons and simultaneously increasing the number of electrons, but we're not adding another electron shell. And so these particular atoms of these elements are very, very electronegative because the valence shell is really close to the nucleus, and so the nucleus has an extremely large polar attraction. And so how do we measure that, or how do we describe that? We call that electronegativity. Because what's in electronegativity? What do you see as a word?
Electron.
Electron, right? And what's the charge of an electron?
Negative.
It's negative. So it's the attraction of the negative electron by the nucleus. And so over here in column 7, even though the right side is stronger than the left, what happens to the valence shells as we go from top to bottom? They get further and further from the nucleus. So even though the nucleus is bigger over here at 85 for AT than for F, those valence electrons are further away, and so the electronegativity of the element AT is lower than anybody above it. And so electronegativity increases going from left to right on the periodic table because the number of protons in a given row increases, and the electronegativity increases as we go from the bottom to the top because the proximity to the nucleus increases the fewer electron shells or orbitals there are. So does that remind you how to do it? Or what it means? And then how to do it is in that lecture assignment or learning assignment where it tells you, the difference, find the difference between the two elements that you're looking at, and then, use the information that's given there to decide if you're going to have a covalent bond or an ionic, an ionic interaction. And if it's a covalent bond, will it be polar covalent or non-polar covalent? And the difference is really how different are those electronegativities from each other, and how able is an atom of a particular element, how well can it attract not only its own electrons, but electrons of anything it bonds to. And that will tell you polar covalent versus non-polar covalent. So that is the really critical stuff from the atomic structure. And in fact, what we really just did is we went through this pretty thoroughly again, okay? The parts for chemical reaction are really limited to, right now, to what was in the PowerPoint about, can you identify this terminology and look at the clues to a chemical reaction. And so if I open that PowerPoint, and I'm not sure if I have to do a new share. Oh, that's my outline right here that I get to finish next. Okay, so let me open that. Did the PowerPoint open for you, or do I need to do a new share?
It did.
It did open? Okay. So you can see here also that I gave you a little bit of atomic history, to explain how did we figure out that there was a nucleus. And so there's a video in the video section about Rutherford and how he figured out that the nucleus was a small, dense, positive set region, and that most of the atom was like empty space with the electrons. So that's something I would suggest you go back and you make sure you know what Rutherford did. And it's outlined here. You could always rewatch that video if you needed to be fresh and just make a note about it. These slides here, I'll cover what I just talked about, the parts of an atom. So, the nucleus and the electron shells and what goes in the nucleus. And it really reviews, how the shells are put together. And then if you have, so for the learning assignments, you had to draw some Lewis dot structure. For the test, you need to be able to recognize correct Lewis dot structure. Since you're not doing a written test for me this term, you're getting off a little bit easy in that sense where you're not having to draw it. You just have to be able to recognize it.
And that's going to be multiple choice, right?
Yeah, it might have a couple like fill in the blank type things where we're looking for some specific answers, but there's not going to be a draw part of this because it's all
I
on
was
the
just wondering
computer.
like how you're going to do the Lewis dot structure, because I don't think I can.
Yeah,
really
it would be recognizing it. Yeah, and
Okay.
like I said, you're only in that sense are you getting off easy because of all the at home learning that you're having to do. So then so make sure you understand how Lewis dot structure shows valence electrons and how you can use valence dot structure to predict what types of bonds are going to form. And so that's why I've got these two slides in here, and I show you where they came from. So it explains, it shows electron dots, and it explains, how the electrons come together, and it shows you, now those shared electrons become that line, so that we know that it's a covalent bond versus a lone pair. And so, remember to refresh Lewis dot structure and understand how it's a tool used to represent valence electrons, so that in conjunction with understanding electronegativity, you can actually show how atoms will form chemical bonds. And the very last part of this was just looking at, so we looked at bond types, and then it was about chemical reactions. And just, I didn't put anything in the learning assignment about this. It's really, do you know what reactants and products are and what the clues to chemical reactions are, because we move forward from this and we talk about enzyme behavior in chapter 4, we're going to be talking about biochemical reactions briefly. And but most of this about chemical reactions, it's just that for you to have a basic conceptual understanding that atoms interact and they form bonds in order to reach stability. They react because they're unstable, because their valence shells aren't full. And that's why we call the elements in that far right column on the periodic table the inert or noble elements, because their valence shells are full, so you don't ever find them bonding with anything else because they're not chemically reactive. So that help kind of pull electronegativity and valence shells and chemical reactions and forming molecules all together for you?
Yes.
Okay, so that was 2.1. So I'm going to close that. And if I pop this guy open again, you know, we've been back through all of this again. And, you know, some of this is just little things like the 4 main elements essential for life. Well, I think you've seen with the biological molecules that we've got carbon, hydrogen, oxygen, and nitrogen making all 4 of those. And so those are the 4 elements. Can I go to 2.2 for the properties of water?
Mhm.
Yeah, and we can go for 10 more minutes, and then I know we didn't start right away because we were waiting to see if people popped in. So I can do an hour. Okay, so for that, we need to go to week 2. And then week 3, all you've got that I copied and pasted the biological molecules learning weekly content objectives back over again, because there was a carryover for you to spend more time on biological molecules, and I didn't make the study guide due until this week, right? It's due on Wednesday for biological molecules, right?
Yeah.
So the study guide that you had to turn in on Saturday was on, oh, this one is actually kind of if you covered this in study guide 2.1, then you wouldn't need to cover it, but I dropped it back in here to remind you that electronegativity really affects bond formation and that it's going to impact how water behaves, okay? So no need to cover it in 2 different study guides, but if you did, then I guess it's in 2 places. So what are the focuses for 2.2? It's looking at water as a polar covalent molecule and how its polarity gives it its specific properties and its ability to form hydrogen bonds, and then looking at how the behavior of hydrogen in solutions, you know, when we drop atoms of elements into water, whether or not the hydrogen ions separate, and if we have excess hydrogen in a solution, then it becomes acidic, and if we have excess hydroxide in a solution compared to hydrogen, then it's basic. So what questions do you have about the properties of water and pH?
I don't understand how to do the pH thing with the log.
Okay, I have kept that pretty simple because there's actually there is a pH of 2.3 and 3.6, but I've just left it whole numbers. And a logarithmic scale means that it is base 10. So I'm going to pop open this slide. Can you see this one on the pH scale?
Yes.
Okay, so pH is really the concentration of hydrogen ions. That's what pH means. And it's done by doing this little calculation called the negative logarithm of the hydrogen ion concentration. And that's where that second bullet says we write it as pH equals negative log, and then in brackets H plus, and the brackets mean literally concentration. And so the pH scale is done from 0 to 14. And I'm going to shift to this one. Does this look familiar? Going from, this one just shows from 1 to 14. It doesn't actually show you from 0. And basically, a solution that has a neutral pH or a pH of 7 has an equal number of hydrogen ions and hydroxide or OH ions. And because it has an equal number, it's neither acidic nor basic, and so we say that's neutral. Now, what happens if there's a shift in the solution based on, adding an acid or, just having a different solution than water altogether? What happens in that solution when we measure how many hydrogen ions there are compared to how many hydroxide ions there are? As we measure, the more hydrogen ions there are compared to hydroxide ions, then the lower the number on the pH scale, but the more acidic. So as the number goes down, the acidity goes up. And as the number goes up to the right, the acidity goes down and alkalinity increases. So why don't we want to come in contact with things like oven cleaner and bleach and hydrochloric acid? It's because either the hydroxide ion concentration or the hydrogen ion concentration of those things is so high that it basically eats through cell membranes, right? So they're really harmful. So how do I calculate it? Well, things with more hydrogen than hydroxide, like are shown here, we call them acidic. And the bigger the difference is, the more acidic, okay? And solutions where hydroxide is greater than hydrogen, we say they're basic. And the more hydroxide compared to hydrogen, the more basic. So what happens if I increase hydrogen ion concentration? I go closer to 0 on the periodic table. And if I increase hydroxide ion concentration, I go closer to 14, okay? And this slide basically shows you that when I'm defining it, I'm looking at how do I get this logarithmic number, the 10 to the minus 14? Why is it from 0 to 14 anyway? And it has to do with a dilution of hydrogen ions. I put a short video in the link with the lab where I wrote a pH scale on the board from 0 to 14, and I explained the molarity of hydrochloric acid, like partway along the table. And I basically said at a pH of 0, you have 1 molar hydrochloric acid, full strength hydrochloric acid. But if I put 1 part hydrochloric acid in 9 parts water, then it's a 1/10 dilution. And so now I have 0.1 molar, where molar just refers to the number of molecules in a solution. And so what happens if I dilute hydrochloric acid more and more and more? What am I doing to the pH of that solution?
Getting small.
I'm making the pH actually closer to 7 because I'm diluting the hydrogen ions, where in a particular volume of water, I would find fewer and fewer free hydrogen ions. And so all that pH is a measure of how many hydrogen ions there are. And in a neutral solution, there's an equal number of hydrogen and hydroxide ions. And so they cancel each other out, and we say we have a neutral solution. So this slide explains how to calculate it. So if I tell you the pH of something right here, and remember pH is hydrogen ions. So if I tell you the hydroxide ion is 10 to the minus 11, you know that the two numbers have to add up to 14. So what plus 11 is 14?
3.
3. So down here it says 10 to the minus 11 and 10 to the minus 3 equal 10 to the minus 3. And all I need to know is that 10 to the minus 3 represents hydrogen ions. And the logarithm equation just tells me take the minus 3, instead of being a superscript 10 to the something, I take it down and I change its charge. I change it from minus to positive, and so the pH of the solution is 3. If I told you the pH is 10 to the minus 6, or the hydrogen ion concentration is 10 to the minus 6, it's the same as saying 0.000001. And that's actually a really small number, but it's more than 6 zeros and a 1 or 8 zeros and a 1. So what's happening is I go from left to right on the pH scale from 0 to 14, I'm diluting hydrogen ions by a factor of 10 every time. So if I look at this here and I have something that's a pH of 12 and I add hydrogen ions to it, if I add 10 times more hydrogen ions than it currently has, its pH will change from 12 to 11. If I add 100 times more hydrogen ions than what it currently has, its pH will change by 10 times 10, 11, 10. Does that help make sense about what's happening with pH? So when,
Sorta.
so if I say I have two solutions and one has a pH of 10 and one has a pH of 6, which one has more hydrogen ions, 6 or 10?
6?
6, yeah, because it's more acidic, so it has more hydrogen. And how many more? Well, every step on the pH scale, because it's a logarithmic scale, is by a factor or a multiplier of 10. So I go from 10 to 9, that's 10 times. 9 to 10, that's 100. 8 to 7, that's 1,000. And 7 to 6, 10,000. So something that is a pH of 6 has 10,000 more hydrogen ions per milliliter of volume than something of a pH of 10. So that's how the scale works. And what it means that it's logarithmic is it doesn't just go from 7 to 8, an increase of 1 in hydroxide. It goes up by a factor of 10 hydroxide and down by a factor of 10. Something that's a pH of 8 versus 7 has 1/10 as many hydrogen ion has 1/10 as many hydrogen ions as 7.
Where is this found? Is this on your
Well, this is the
study guide?
PowerPoint. This is the PowerPoint,
For
right?
which one?
2.2 water and
2.
pH. Yeah.
Okay.
So that's been in there the whole time. And then there are videos for you to watch. And then if you go back to the videos that I posted to do with the lab, there's a short video, like 5 minutes, where I explain the scale on the board. Okay?
Thank
All
you.
right, you're welcome. And then properties of water, you need to know how those work. And there are tons of videos in there that go over how that works. You need to understand hydrogen bonding and how that works. And again, there are videos that I made for the lab that explain the hydrogen bonding and that it's a temporary association because of the uneven distribution of electrons. So if you're struggling with that, I really recommend go back and watch a couple of videos and try to make sense of it some more there. And then with the biological macromolecules, it's really, I spent a bunch of time in a Zoom session talking about carbohydrates and lipids and proteins, and I only spent like 5 minutes on nucleic acids, but there were also many, many fewer slides on nucleic acids. And I'm going to go back to my modules here. And hopefully you noticed, and you may not have watched them yet, I popped them in at the end of week 2. I popped them in after. See here also, Lab Module 2 Gregor demo videos. So those are where those are for pH and hydrogen bonding. And so those are helpful not only for the lab, but also for understanding how water behaves for the test, right? And then these videos on biological molecules, there's Bozeman Science does an overview, Amoeba Sisters does an overview, and then I put in these ones where you've got carbohydrates, protein structure, and nucleic acids all together. And I didn't add one on lipids. I had covered that pretty thoroughly, but it wouldn't be that hard for me to find one on lipids and throw it in here. But lipids is covered within these first 2 that cover all 4 together. And so I would recommend watching those. Were there any questions left about biological molecules?
I kind of struggle with the the DNA and RNA?
We are covering DNA and RNA in detail in chapter 9. What
Mhm.
I need you to know is that DNA and RNA are both nucleic acids, and that both of them have a repeating polymer structure where the monomer is one of 5 nitrogenous bases, a 5-carbon sugar, and a phosphate group. And the thing that makes DNA different from RNA is which 5-carbon sugar it is and which of the bases. DNA uses 4 of the 5 bases and RNA uses 4 of the 5 bases. And DNA is double-stranded and RNA is single-stranded. So those are the key pieces. I don't need you to memorize the structure of pyrimidines versus purines. I did talk about it in the Zoom that we did on this already, but I explained that I didn't need you to memorize like what cytosine looks like. I just need you to know that these names refer to the nitrogenous bases that are found in DNA and or RNA. And can you see how this slide shows you that thymine is in DNA only and uracil is in RNA only? We are going to talk about how DNA replicates in chapter 9, and we're going to talk about how RNA is used as a temporary copy of DNA to make proteins. And for now, all I want you to understand is that in a eukaryote, something with a nucleus, the DNA stays in the nucleus, that RNA is made as a copy, and that it leaves the nucleus, and that outside of the nucleus is where proteins are made. So RNA is the copy of DNA in a eukaryote that is used to make proteins outside of the nucleus. There is actually some protein synthesis that happens in the nucleus, but that's more detail for another time. And then slide 46 shows you, and I covered this in the other Zoom meeting, but just a reminder, see these 5-sided pentagons here, and then the blue dots, and then the 5-sided red pentagons and the red dots. The red and the blue pentagons represent the sugar molecules, and the blue and the red dots are phosphates, and the phosphates are all the same, and the sugar molecules are all the same in DNA. It's just they're showing blue and red to show you that DNA is two-stranded, double-stranded, and that what makes it able to bond together, it's the hydrogen bonds in the nitrogenous bases. And the way that DNA carries a code is in the sequence of its bases, the order in which they are creating a polymer. And so where do our genes come from? Our genes are lengths of DNA that code for proteins, and what particular gene I might be talking about is determined by the sequence of those nitrogenous bases that are oriented here in the middle. And what do these dashed lines represent? Those are the hydrogen bonds. So it's not covalent bonds holding the 2 strands together. It's millions and millions of hydrogen bonds between the 2 strands. All right, are we good?
Thanks.
You're welcome. And in fact, slide 47 talks about hydrogen bonds in DNA. And then I think that about covers it. And this explains, you know, a little bit about the structure of DNA. And you don't need to know more about DNA than what is covered in these few slides here. You will learn a whole lot more about DNA than maybe you cared to know, other than it's awesome, when we cover chapter 9. Okay, this is just to get you understanding that DNA is in the biological macromolecule category of nucleic acids. So when you're working on the test, you'll have access to your study guides, to the videos, to the PowerPoint. But if you don't understand the stuff and know where to look, then you won't have time to look it up and to answer every question. That's why you need to study and why you need to know this stuff, and why having a study guide out where you have very specific organized information will help you to locate something that you sort of remember. And I think that wraps it up for what we were gonna cover. And so I stopped sharing and it's just you, Marie.
Hi. No wonder it got quiet.